Hello everybody, I am Deepika from LearnoHub, a free learning platform where you can study physics, chemistry, maths, biology absolutely for free only at learnohub.com. So students, now we are going to start with our next chapter and the next unit, that is human physiology. So this unit is all about human circulatory system, nervous system and many more, right? So this is a very interesting unit and I hope you'll understand it very well through our videos. So starting with our first chapter of human physiology, that is the circulatory system. So let's get started. So while studying about different fluids, while talking about different animals, while talking about different plants, we have studied that there are different fluids that circulate inside a living organisms. And what do these fluids do? So they perform different kinds of functions, right, in different organisms. Some of them, they carry substances from one place to another. Some of them carry substances to provide nutrition. Some of them carry substances for you can say for excretion. Some of them transport the substances so as to provide immunity in the body, right? So this is what we are going to study. So our first topic today is the need for transport inside the body. So what is the need of this transport inside the body? So there is a fluid which actually transports the substances inside the human body. The first is the digestive system. So the reason why there is a need of transport, so we know that when we eat food, our body digests it. Our small intestine digest all the food and absorb this food, right? So now this food nutrients, they are absorbed inside our small intestine, but they are not just needed in the intestine, but in the different parts of our body, right? So our brain needs some of the nutrients, our muscles need the nutrients, our other parts of the body, they need nutrients, right? So as to grow well, so as to repair. So there are many functions, correct? So the digest and absorbed nutrients which need to be transported to every cell of the body is done by these fluids that are present inside our body. The second is respiratory system. So we know that we inhale air, right? So when we inhale a lot of oxygen, and this oxygen is very much required by our body cells so that they can release energy, they can perform their function and they can release energy from our food from our cells, right? So for that, who actually takes this energy? So there is a fluid inside our body which actually take this oxygen to our cells, and in turn when respiration happens, carbon dioxide is released by the cell, which is a byproduct which is very toxic to our body. So that also need to be transported back to our lungs so that our body can get rid of that toxic substance, that is carbon dioxide. Clear? So when we inhale and pick oxygen, when we inhale, so this fluid actually picks oxygen in the lungs, which is needed by all the parts of the body. So carbon dioxide is collected from entire body and it is carried back to the lungs to exhale out of the body. So when we inhale, we are taking in a lot of oxygen which is needed by our cell. And now my body cells have already used this oxygen, and now they have, what do? They will release carbon dioxide. So when I exhale, so at that particular time, that carbon dioxide is given out of my body. Clear? The next point is excretory system. So there is excess water, there is excess salts, and there are excess nitrogenous waste, that is the urea, which needs to be removed from different parts of the body. So who does that? So there is a fluid which carries all these toxic, all these extra substances from the body for excretion. Clear? The next is hormones. Now the hormones, they are produced at a point, at a particular organ or a gland, and now they are not needed at that gland, but they need to be transported to the target organ, right? So who takes those hormones to the target organs? So again a fluid inside our body, right? So they are secreted by endocrine system and they have to be carried throughout the body by the circulating blood, which to act wherever they are required. So wherever these hormones are required, who carries these hormones to that target organs? So that is done by the body fluids, correct? So I hope this much is clear. Now the next is the fluids in our body. So basically the two main fluids which help in the transportation of the body nutrients, body waste, or the other, which actually perform the function of transport. So they are blood and lymph. So who are they? So they are blood and lymph, right? Now there are actually three principal fluids. How many principal fluids are there? There are three principal fluids. So one is blood, tissue fluid, and the lymph, right? So we will study all of them in details. First of all, we'll just find out where do we find these fluids. So blood is actually present in our heart and in the blood vessels. So what are these blood vessels? So blood vessels are arteries, veins, and capillaries. So the blood is majorly contained in heart and these blood vessels, right? Then coming on to tissue fluids. So tissue fluid is present between the spaces of the cells. So there are different cells and these different cells have certain spaces inside them. So the occupying spaces between the cells of the organs. So inside organs are made up of cells, right? So these cells have certain spaces between them. So this is the place where we find tissue fluids. Correct. The next one is lymph. Where do we find this lymph? So lymph is contained within lymph vessels and lymphatic organs such as spleen and tonsils. So they are present in spleen and they are present in tonsils. Okay. So I hope this topic is also clear. So what are the three major principal fluids in our body? So they are blood, tissue fluid, and the lymph. So we'll talk about all of them separately. So they just not only transport the things or the nutrients inside our body, but they have many other functions to perform, which we will discuss in our coming sessions, right, in our coming slides. Now there are certain fluids which are not circulating. So they are basically present in a particular organ. So all of them they are said to be the circulating fluids, but there are certain non-circulating fluids, right? So these non-circulating fluids. So first one is the synovial fluid. So this is the fluid which is filled inside the cavities of our skeleton joints, right? Now there is another fluid that is vitreous humor, right? So where is it present? It is present in our eyes. So these are some non-circulating fluids that are present in our body that are confined to a particular area, right? So they do not move out of that area. So I hope this is clear. Now let us take a look at the diagrammatic representation of blood and lymph circulation. So blood flows in heart and blood vessels, whereas lymph flows in the lymphatic system. So that includes these lymph capillaries and the lymph nodes and the lymph organs, right? So as you can see, these are the lungs, right? So from lungs, what do this blood get? So we get oxygen. Our blood get oxygen from the lungs, right? So this then goes to our heart. From heart, it goes to the different parts of the body, and in different parts of the body, this oxygen gets used up in respiration and carbon dioxide is given out. Right? Now the carbon dioxide is given back to the heart. From heart it goes back to our lungs, right? So this is a complete circulation of blood inside our circulatory system, right? From heart to blood vessels and then back to lungs, right? So this is how it happens. In case of lymph, this lymph, it is basically a white, or you can say a pale white colored fluid which travels inside the lymphatic capillaries, inside the lymph nodes or the lymph organs, right? So there is a lymphatic system where this lymph flows, and inside over here you can see the tissue fluid. So this tissue fluid is basically present inside the spaces of the cells. So there are different cells, and inside those cells there are certain spaces. So this lymph basically, the tissue fluid is basically present between spaces of these cells. So I hope this diagrammatic representation of blood and lymph circulation is clear to you. Now, the type of blood circulation. So as we have seen, if we talk about blood, so blood flows from heart to different parts of the body and then back to the heart, right? So it is basically flowing inside heart and then inside the blood vessels. Correct? So this is what we have seen. So this type of blood circulation is called as a closed blood circulation, where the blood circulates in a closed manner inside certain vessels or certain organs. Correct? So inside the blood vessels, for example, in case of human beings. But if we observe the blood circulation in certain insects, so we will see that the blood mostly flows through the open spaces in the body. For example, insects. So the blood flows from heart to body spaces without blood vessels, right? So blood is present in the heart, so then it flows to the body spaces. So inside the spaces in the body, right? So there are no blood vessels to carry this blood from heart to different body parts, right? So this type of blood circulation is called as open blood circulatory system, where the blood flows in the open spaces of the body. I hope this is clear. Now, the properties of blood. So you must have seen blood, your own blood, or your friend or your family member might have experienced a small cut on the body, right? So you must have seen the blood coming out from that cut part, correct? So everybody has seen blood. So it is very simple for us to understand the properties of the blood. So blood is never stationary. So as soon as you see the part of your body has been cut, so you see the blood flowing. Blood is coming out. So this means that blood is not stationary. So it is continuously moving. So that is true. The blood is never stationary and it is continuously moving inside our body. Correct? The second thing is the color of the blood. So what do you think? What is the color of the blood? So whenever we draw a diagram, we always use red color for oxygenated blood, that is oxygen rich blood, and we always use blue color for dark blue color for deoxygenated blood or carbon dioxide rich blood, right? So it is not actually very true. So the color of the blood is the red color, the blood which is actually flowing in the arteries, or you can say the blood which is oxygen rich, is bright red in color, and the blood which is in veins, mostly the deoxygenated blood. So there are exceptions that we'll study. So that blood, deoxygenated blood, is dark red in color. Correct? So there are two colors, bright red and dark red. So bright red when the blood is in arteries, and dark red when the blood is in veins. Correct? Now the volume. If we talk about the volume of the blood, so the blood, if we talk about an average adult, if you talk about yourself now, okay, so you are little small, but an average adult human being has 5 to 6 liters of blood by volume. Correct? Now talking about the taste. So have you ever tasted blood? You must have done it in your lifetime. So you must have experienced a cut, and what do? You immediately, so if there is a cut on your finger, so blood starts coming out. So what is the first reaction? So mostly what will a person do? So person will put that finger inside the mouth, right? So this means you have tasted your blood, right? So what is the taste of the blood? So it is slight saltish, right? So the pH of the blood is 7.3 to 7.5. So it is slightly alkaline. 7 is the neutral one, right? So it means it is neither acidic nor basic. But if it is more than 7, so it means it is alkaline. So in this case, we can say the blood is slightly alkaline. Clear? So 7.3 to 7.5 is the pH of the blood. Now talking about the functions of the blood. So what does this blood performs? What are the functions that a blood performs? So there are two main functions. The first function is transport. So transport of oxygen, transport of carbon dioxide, transport of other waste substances inside our body, right? And what about the protection? So blood has certain cells which helps in the protection of our body, right? Which protects us from various diseases. So what we are going to do is, now we are going to talk about each of these functions in detail. So let's start with the functions of blood. Now let us start with the first function of blood, that is the transport by blood, right? So the transport by blood. So there are different transports that happen through blood. The first is transport of digested food. So as we all know that when we eat food, it goes through our alimentary canal, and when the food reaches small intestine, so the digestion of carbohydrates, fats, proteins, all of them are completed in the small intestine itself, right? So it is the place where the absorption of these nutrients takes place, right? So the food that is digested is absorbed in small intestine only. So these absorbed nutrients, they are basically needed by different tissues of our body. So who helps our body to distribute these nutrients? So these can be simple sugars like glucose, amino acids, vitamins and mineral salts. So all of them, they are carried from small intestine to different parts of the body. So what do? They help in the transport of the digested food. The second one is transport of oxygen. So as I was telling you, when we inhale, right, so we take in a lot of oxygen, so it goes into our respiratory tract. So there is a connection between our respiratory tract and the circulatory system as well. So what happens, like in the first case, our circulatory system and our digestive system, they were somewhere meeting, right? So same is the case in with the second transport also, where the oxygen goes into our lungs. So in lungs, there are small, tiny sac-like structures, like so balloon-like structures, which are called as alveoli. So they have very thin wall, which allows diffusion of gases. So over here, there are many blood vessels which are surrounding these alveoli. So when blood comes to the lungs, so in lungs there is the blood is actually full of carbon dioxide, the gas which is released by our tissues, right, while respiration. So this blood is rich in carbon dioxide, but the alveoli, they are rich in oxygen. And now there is affinity of oxygen, which affinity of a component that is called as hemoglobin. So there is a component called hemoglobin which is present inside our body, inside our blood, right? So this blood has more affinity for oxygen as compared to carbon dioxide. So as soon as this blood, it reaches this part, the alveoli, the blood, it is in the blood vessels near the alveoli. So now, since hemoglobin has more affinity to oxygen, so the blood attracts more and more of oxygen from the alveoli, and it gives away the carbon dioxide to the alveoli. So what will happen, when we exhale, this carbon dioxide will go out of our body, and the blood over here will be rich in oxygen, and now this blood will go back to our heart for circulation of this oxygen rich blood into our body. Correct? So what will happen, the transport of oxygen from lungs to tissues by means of RBCs. So where is this pigment hemoglobin present? So it is basically present in the red blood cells, which we'll study in our further slides. So there are different cells, and one of them is RBCs, which contain hemoglobin, which has more affinity for oxygen as compared to carbon dioxide. So it picks up oxygen and it gives away carbon dioxide. So it forms an unstable compound called oxyhemoglobin, right? So there is hemoglobin, it forms an unstable compound with oxygen, that is HbO2, that is unstable compound which is oxyhemoglobin. Correct? So this is how it transports oxygen in our body. The third function is transport of carbon dioxide, which we almost studied in point 2 also, right? So from tissues to lungs. So what happens? So oxygen rich blood will go back from lungs, right? So these are the lungs. So it will go back to the heart, one side of the heart, and from heart this oxygen rich blood will go to the body tissues. So as soon as it reaches the blood tissues, what will happen? Now the oxygen will be picked up by the tissues, and when they will perform their functions, when the respiration will occur, there will be a lot of carbon dioxide that is released by the tissues, right? So these blood vessels will deliver oxygen to those tissues, and in turn they will take what? They will take carbon dioxide, right? Now this carbon dioxide will be taken back to the heart, and from heart it is given back to the lungs for oxygenation. So it basically transport carbon dioxide from tissues to lungs, right? So partly the transport of carbon dioxide happens with hemoglobin, and partly it is in the solution form, right? So it makes a solution with our blood plasma, and partly the carbon dioxide is transported in that form. Clear? So hemoglobin plus carbon dioxide, so they also make a very unstable compound, that is HbCO2. Clear? So this is how carbon dioxide is transported. So there is only one way the oxygen is transported, that is it will make an unstable compound with hemoglobin and it will travel in our blood vessels, right? But in case of carbon dioxide, so carbon dioxide is traveled, one, by making an unstable compound with hemoglobin, right? And secondly, in that solution form with our blood plasma, right? So this is something important which you must remember. Correct? So let's see the next function of our blood as a transport system. Now the fourth function is transport of excretory material, as I told you. So tissues, from tissues to liver, kidneys and skin. So whatever is the waste product that will be transported to our various body parts like liver, our excretory parts of the body like liver, kidney and skin, right? So they are eliminated from our body till or they are made harmless for our body. The second one is distribution of hormones. So these hormones, they are made inside our body in certain glands. So they are called as endocrine glands, which we call them as ductless glands, right? So they do not have any ducts. So they send these hormones which they produce into the blood. So just Simran ji le apni zindagi, right? So these hormones are Simran, so they are allowed to go with the blood and they are allowed to go to their target organs wherever they want to go. Clear? The next one is distribution of heat. So this is also one of the very important function, that blood helps in keeping the temperature of the body uniform by distributing heat. So this blood helps in distributing the heat throughout our body, and hence our body temperature is made constant by blood. Now the next main function of our blood is protection, right? So how does blood helps in protecting us, right? So you must have seen when there is a cut, there is a cut in the blood vessel, so we see blood coming out, right? But after some time there is a formation of some hard substance which is called as clot. So what is this clot? This clot is formed by blood itself. There are cells inside our blood which forms this clot, right? So you must have heard somewhere that in an accident a person lost so much of blood that that person finally collapsed, right? So what is the reason? Because we know that blood performs a very important roles in our body of transportation, right? So if we need a certain amount of blood, we need the blood to flow in certain way so as to perform our body function, so as to transport the substances which are needed in different parts of the body, right? So blood heals itself in some way. So there are cells which are present in the blood, they form clot which prevents the further loss of blood, right? And also when there is a cut, So there is a chance that microorganisms will enter into our body and they might cause some or the other infection. But when the clot is formed, so this clot prevents the entry of the disease causing microorganisms, right? So it is always said that keep your hands away, don't touch anything, right? Because the microbes can be anywhere which can enter into your body, which can create certain infections in your body. So this is how blood forms clot, and this is how this clotting is helpful to us, right? It helps in protecting our body. The second function is white corpuscles. As we were studying in functions, in the transportation functions, that there are there are RBCs which contain hemoglobin, which helps in transport of oxygen and carbon dioxide. In the same way, there are some other cells in the body which are called as WBCs, white blood corpuscles, which protect our body from diseases by engulfing bacteria which may have entered into our body, right? So it might happen that certain pathogens enter our body in some or the way other, either with the food, or you can say from any cut part, right? From any if there is any cut, the microorganism may enter, or inhalation. So there are many ways how the microorganisms can enter into our body. So there are white blood cells which actually engulf these bacteria. They eat these bacteria and hence they protect our body from the infections. Third is they produce antitoxins and antibodies which neutralize the poisonous substances or kill the germs which have entered into the body. So there is another role of our blood, that there are certain cells in our blood which helps in producing antibodies. Antibodies, these are friends of our body. These are basically protect our body from various kinds of germs and various kinds of poisonous substances. Clear? So I hope the protection functions of our blood is clear to everyone. Now the composition of our blood. Now our blood contains, the blood is basically what? It is a fluid, right? So it means there is some fluid part, right? And there is some non-fluid part, right? So this is basically what? This is blood. Clear? So there is a fluid part of blood which is called as plasma. Clear? So what it is called? It is called as plasma, and it constitutes the maximum percentage of blood. So that is 55 to 60% of our blood is plasma. Clear? Then there is a non-fluid part. So what is this non-fluid part? So those are the cells. What are these cells? So these cells are RBCs. So we have already studied they have a very important function in transportation of oxygen and carbon dioxide in our body. Then there are WBCs, white blood cells or white blood corpuscles, which have very important function in protecting our body against various pathogens, right? Then there are platelets. So these platelets also we have studied where in blood clotting, in the formation of clot. So when blood forms clot, so it is only possible because of the platelets. So platelets are the cells which helps blood in the formation of clot and prevents the further loss of blood. So I hope this is clear. So also the cellular part is around 40 to 45% and rest 55 to 60% is just the plasma. Right? So I hope this is clear. Now let us talk about the first part of our blood, that is the liquid portion or the fluid portion of our blood, that is plasma, which constitutes 55 to 60% of our blood. So how does it look like? What all does a plasma contain? So let's talk about it. So the first thing is it is light yellow colored fluid which is alkaline liquid. So it is alkaline in nature. It is not acidic, it is not neutral, but it is towards the alkaline side, right? So the first thing that it contains water. Because of water it is fluid, right? So 90 to 92% of the plasma is water. Rest 7 to 8% are the proteins. Then there are certain inorganic salts, which is like 1% of the plasma is inorganic salts. For example, sodium chloride, sodium bicarbonate. Then there are some other substances which you will see in blood. The first thing is glucose, amino acids, fibrinogen, hormones, urea. So all of them are in trace amounts, like very, very minute quantity. If we talk about plasma, and if we remove these proteins from plasma, right? So these are fibrinogen, these are the proteins which are present inside the plasma, right? So if we subtract plasma from proteins, if we separate out plasma and proteins, right? So if we take out the proteins from plasma, so what we will call it? We'll give it another term, that is serum. So serum is plasma minus proteins, right? So there will be water, there will be inorganic salts, there will be other substances in trace amounts, but there will be no proteins in serum. Correct? So I hope this is clear. This composition is very important to understand. This composition is very important. So there might be like 1 mark questions from these small topics in your exams. Now talking about the second part of blood, that is the cellular elements or the cellular components or the formed elements of our blood. So they are not the fluid part, they are the non-fluid parts, right? So what are those? Those are the cells which are present inside our blood. So you will see 3 major categories. One is RBCs, that is the red blood cells, right? So these are the RBCs, which are also called as erythrocytes. Clear? Second one is white blood cells, which we can write WBCs, right? And we also call them as leukocytes. Clear? The third one is blood platelets, which we call as thrombocytes. So these are basically what? These are the platelets. So now in detail we will discuss each of them. So the first thing is, if we talk about the red blood cells, so red blood cells look something like this, right? So they have, there are only one type of red blood cells, and we know that what do they contain? They contain hemoglobin, which is very important in the transportation of gases, right? But if we talk about white blood cells, so white blood cells are of different types. There are different kinds of white blood cells: neutrophils, eosinophils, basophils, lymphocytes and monocytes. So there are different categories of white blood cell which we will study in detail. What are the functions, how do they look, what is their structure? So all of them that is needed in our grade, so we'll study each of them. And also there is one more thing, that is third type of cells, that is the platelets. So where we have studied platelets, just recall. So we have studied them in clot formation, right? And where we have studied the white blood cells. So white blood cells we have studied in the protection functions of our body, right? So let us look into the detail of each one of them. Now starting with red blood cells, we call them as RBCs, or we also call them as erythrocytes. So these are the oxygen carriers of our blood. So how do they look? What is the shape of RBCs? So they have minute biconvex disc-like structures. As you can see, they have this disc-like structures which are basically flattened from the center, right? You can see this cavity over here. So they are flattened from the center, but on the outer side, they are round and they are thick. Clear? Then talking about their size, so they are 7 micron in diameter. So their diameter is 7 micron. The next is small size plus concavities on either side provide a large surface area which makes them very efficient in absorbing oxygen. Now, if we talk about their shape, so they are very small, plus they have concavities on the either sides, which actually gives large surface area to them where they can actually absorb oxygen and they can transport oxygen. Then the small size enables the RBCs. Now because they are very small in size, so they can even travel through those minute capillaries. Capillaries have very small size, right? So they can even travel through very fine capillaries in the body. Correct? So I hope this is clear. So if you must have, your family must have gone through a blood test, right? So you must have seen the report where on the sides it is written how much is required. If you talk about RBCs, if you see the RBC count, so it is written differently for male and female. So for males the RBC count is always written higher, and for females it is always written little less, right? So let us talk about what is that count, right? So if you talk about a normal adult male, right? So for them the number of RBCs that they have is 5 million RBCs per millimeter of blood, right? If we talk about females, adult human female, so it is 4.5 million RBCs per millimeter of blood, right? So this is the amount, or this is the number of RBCs that are needed in that are actually there in a normal adult male and female, right? So just next time whenever any person is having a blood test from your house or from your family, just look into the blood reports very carefully. So just see the normal ranges, so they are different from males and females, and just look how much RBCs do they have in their body, right? So just observe all those things in the reports. So let's see what is next about RBCs. Now talking about RBCs, and in RBCs, let us talk about hemoglobin. So we know that in RBCs, they contain a pigment which is called as hemoglobin, which is very, very essential pigment that is present inside the RBCs because they are the carrier of oxygen and carbon dioxide. So it is a very effective chemical constituent of RBCs. So it is colorless, spongy body or stroma which contains a respiratory pigment called hemoglobin. So inside RBCs, there is a spongy body or there is a stroma which contains this hemoglobin. Now, the iron containing part, so this is the iron containing part which is called as hemin, and there is a protein part which is called as globin. So basically, hemoglobin has two parts. One is iron containing part and another one is protein containing part, right? So the iron containing part is hemin, right? And the protein containing part is globin, right? So I hope this much is clear. Now the function of this hemoglobin is very important. It combines readily with the oxygen, so it has very strong affinity for oxygen, and it forms an unstable compound, that is oxyhemoglobin, which it readily gives when it reaches the tissue. So as soon as the oxygen rich blood reaches the tissues, so those tissues are very much in need of oxygen, right? So as soon as the blood reaches the tissues, it gives up the oxygen to those needy tissues, right? Then they carry very small quantity of carbon dioxide in form of carbaminohemoglobin. So it is HbCO2. So this is carbaminohemoglobin. So in this form, it carries carbon dioxide also, but very less carbon dioxide. Rest of the carbon dioxide is dissolved in plasma. So it is transported when it forms a solution with plasma. Clear? So I hope the functions of hemoglobin are clear to you. Now, like we always talk about, hemoglobin has more affinity to oxygen as compared to carbon dioxide. But there is another gas for which the affinity of hemoglobin is very high. And what gas is that? So that is carbon monoxide, that is CO, right? So if there is carbon monoxide present around our the place where we are, if the carbon monoxide is more. So hemoglobin has the strong affinity for this carbon monoxide and it attracts the carbon monoxide. If we inhale, basically if we inhale the carbon monoxide, so it will form an unstable product with, not unstable but actually a stable product with hemoglobin. So carbon monoxide and hemoglobin will form a stable product which is called as carboxyhemoglobin. And how do we write it? It is HbCO, right? So this is carboxyhemoglobin. Now it reduces the oxygen carrying capacity of our blood. Why? Because the RBCs have hemoglobin which carry oxygen. Now this hemoglobin is already loaded with carbon monoxide, so there is no place for oxygen to carry, right? So all the surface area of hemoglobin is covered with what? RBCs is covered with carbon monoxide, so there is no place for oxygen to travel along, right? So it cuts down blood's capacity to transport oxygen, and this sometimes leads to death, and this is given another name which is carbon monoxide poisoning, right? So important one. So you must have heard or you must have read in a newspaper that a person died because of the carbon monoxide poisoning. So what happens in hilly areas, because it is very cold, so they burn woods near their say in their rooms, right? So what happens if there is no ventilation, so that when the burning woods will actually release carbon monoxide, and if a person sleep in that room with that burning, you call it as angithi, right? So if a person sleep in that room, so the carbon monoxide is gradually, the concentration of carbon monoxide is increasing. There is no cross ventilation, there is no air coming inside and going out, so basically there is no cross ventilation, right? So in that case, the person's body will inhale, the person will actually inhale carbon monoxide, and finally what will happen, all the hemoglobin will get loaded with carbon monoxide, and there will be no place for oxygen to get transported through that hemoglobin, and finally that person will die because of the carbon monoxide poisoning, right? So sleeping near burning furnaces in small rooms without ventilation can result in carbon monoxide poisoning, which will lead ultimately to death. So it is very important, if you are burning something, if you are in hilly areas and say if you are burning some furnace, or you can say angithi in your room, so it is very important that you must keep a window or door or anything like slightly open, so that the air can come inside and the carbon monoxide air can actually go out, so that does not that carbon monoxide should not enter into your body, and there should be oxygen around you, so that oxygen you can actually inhale, right? So I hope this is also clear to everyone. Now talking about the RBCs, their life and their death. So first of all, let's talk about where are they produced. So in case of adults, RBCs are produced in the marrow of long bones, especially in ribs, breastbone, ileum of hip girdle. But in case of embryos, the RBCs, they are produced in liver and spleen, but not in the bone marrows, right? In case of children until the age of 5, the RBCs are produced in bone marrow of all the bones. In all the bone marrows, in bone marrows of all the bones, RBCs are produced till the age of 5. Then the mature RBCs have no nuclei. Now when RBCs actually mature, they have no nuclei, which when they are produced, they have one. So when the RBCs are actually produced in the bone marrows, they have one nucleus, but on maturation they lose this nucleus and they become enucleated, right? So E, without nucleus. Clear? Then the average life of RBCs is about 120 days. So what is the average life of RBCs? How long do they live? So they live just for 120 days. Now old and weak RBCs are destroyed by spleen, liver and bone marrow. So where do these old and dead RBCs go? So they go back to liver, spleen and bone marrows. Clear? So I hope this much is clear. So if we talk about normal adults, so there are approximately 2 million RBCs are destroyed every second. So every second, in 1 second, almost 2 million RBCs are destroyed in normal adult. Okay. Now let us talk something more about the mammalian RBCs. So we know that when RBCs are produced, they have one nucleus, but as they mature, they become enucleate, right? So they become enucleated. So they actually lose nucleus. Clear? So when the mature RBCs circulate in the bloodstream, they are devoid of certain organelles. The first organelle that we have already studied is they do not have any nucleus. Secondly, they do not have any mitochondria. And thirdly, they do not have any endoplasmic reticulum. Clear? So they are devoid of these organelles, and this though they are deficient in these organelles, but still they are more efficient and they can perform their functions more efficiently. So they can effectively perform their task even though they are devoid of nucleus, mitochondria and endoplasmic reticulum. Still they are able to perform their functions very efficiently. So let's see how do they do it. So what happens when there is no nucleus? So how does that helps in efficient oxygen transport? So as you can see, these are the immature RBCs, right? So they are the newly formed RBCs. These are the immature RBCs. So as they mature, so they look something like this. So these are the mature RBCs. So what is the difference between the two? The first difference is they do not have any nucleus, right? So when they lose their nucleus, it makes them biconcave. So it makes them biconcave, increasing the surface area volume ratio for absorbing more oxygen, so that they can absorb more and more oxygen. Secondly, the spaces in between increased. The space in between them increases more, right? So now this space which was occupied by this nucleus is empty now. So the space has increased. Now the more RBCs can accommodate in the same place. So at the same place, more and more of RBCs can be accommodated, right? So firstly, the shape becomes biconcave, which increases the surface area for absorbing oxygen. Now in a small space, right? So now firstly, this space has of oxygen, the space where the nucleus was present, so that nucleus has been removed. Now they are more thin, they are more lean, which actually accommodates more RBCs. So over here you can see this much space is consumed by 3 RBCs, but now they have become more thin, more lean. So more RBCs can be accommodated in this place, right? So you can draw some more. So that is what it is written. The space in between increased and more RBCs can be accommodated in the same place. So a place where there could be 10 RBCs because of the nucleus, now in that place we can accommodate say 20, right? So 10 more RBCs can be accommodated, which means more oxygen can be transported. Clear? So I hope why the loss of nucleus makes mammalian RBCs more efficient is clear. Now the second one is loss of mitochondria. How this loss of mitochondria makes these RBCs more efficient. So we know that there is cellular respiration. So just recall the functions of mitochondria, right? So cellular respiration occurs in mitochondria, which means mitochondria requires oxygen to perform its functions, right? And secondly, there is glucose which is metabolized, right? So for this also like the glucose is not used by mitochondria, right? So it also requires glucose. Clear? So when there is no mitochondria in RBCs, so RBCs cannot use oxygen for themselves. So still they do not have mitochondria, RBCs do not have mitochondria, so they do not need oxygen for themselves. So RBCs do not need oxygen for themselves, firstly. So all the oxygen that is absorbed from the lungs will be transported and delivered to the tissues, right? Then loss of mitochondria means full transport of glucose in blood plasma unused by RBCs. So RBCs will not use any of the glucose that is present in the plasma. So that will be fully delivered to the other parts of the body, right? So RBCs will not use any of the glucose that is present in blood plasma. Clear? So I hope this is also clear. So now, no endoplasmic reticulum. So now if you talk about the structure of the cell, so this is a normal cell, right? So it has a nucleus, it has endoplasmic reticulum, right? So this endoplasmic reticulum is so much spread inside the cell, right? So when this endoplasmic reticulum is absent, so it makes RBCs more flexible and they can actually pass through the narrow capillaries. Now the capillaries, they have a very narrow space, right? So to pass through it, they have to be very flexible. So when this endoplasmic reticulum is absent, so it makes the RBCs, it increases the flexibility of RBCs to pass through those narrow capillaries. Clear? So I hope it is very much clear why the absence of nucleus, mitochondria and endoplasmic reticulum makes RBCs more efficient. So there is some more information about RBCs which I think you must know. So the first is the newborn infants, they have large number of RBCs. So that is 6.7 million per cubic millimeter, right? The next one is RBC count is lowered 5% during sleep. So when you sleep, so after you get up, 5% of the RBCs are removed from your body. Then the RBC count is higher during physical activity. So when you run, when you play, so at that time the RBC count get increased. During pregnancy also it gets increased, and during emotional upsets also, so this RBC count gets increased, right? Then people living at a height of 4200 meters, so there are people who are actually at a height at around 4200 meter above the sea level, so and above, right? So there is increase in RBCs by 30%, so they need more and more oxygen, right? So they need more and more RBCs. So the RBC count gets increased when a person is at 4200 meter above the sea level or more than that. Then there are two very important terms. First is polycythemia and erythropenia, right? So polycythemia means there is abnormally increased number of RBCs. Erythropenia means abnormal decrease in number. So remember E means removal, right? So just talk about E, okay? So just think about E over here. So it is the removal, that is the decrease in number of RBCs, right? And polycythemia, there is abnormal increase in number of RBCs, right? So I hope this much is clear. So now talking about the second type of cells in our body, that is WBCs, white blood cells. So they are also called as leukocytes. And how do they differ from RBCs? So WBCs have nucleus, which RBCs don't have, right? But RBCs have hemoglobin pigment, which WBCs don't have. Clear? So this is the point of difference between RBCs and WBCs. Clear? The next thing is the number is much less as compared to RBCs. So this is another point of difference between RBCs and WBCs. So just recall, in a normal human adult male, they have around 5 million RBCs per cubic millimeter of blood. right? And if we talk about female, so it is 4.5 million, correct? But if you talk about WBCs, so they are very less in number, so that is 4,000 to 8,000 per millimeter cube or per cubic millimeter of blood. Clear? Now, there is one very important term and the question sometimes come in your exams also. What is this diapedesis, right? So these RBCs, they do not have any fixed shape as amoeba. You remember amoeba, that very funny creature which changes its shape and which forms a pseudopodia to engulf food, right? So the same funny behavior is shown by WBCs also, where most WBCs are amoeboid and they can produce pseudopodia with which they can squeeze through the walls of the capillaries into the tissues. Now they need to go into the capillaries. Now we know that capillaries have very thin walls, right? And they have very small area. They are very narrow in size. So if these WBCs, they want to go into those capillaries, they form this pseudopodia, as you can see in this structure. So they form this pseudopodia and they get squeezed into those capillaries, and for this the term given is diapedesis. Clear? So I hope this term is clear to everyone. Now the type of white blood cells, that is WBCs. So based on the shape and the other characteristics, these WBCs have been divided or classified into two types, right? So the first one is granular WBCs and another one is non-granular. So it is very clear from the names only that the granular WBCs contains some granules, right? So the cytoplasm contains granules, whereas the nucleus is usually constricted in the lobes, right? So nucleus is constricted in lobes. So there are certain lobes that can be seen in the nucleus. Clear? So what are the examples of these granular WBCs? So when we were taking an overview of these blood cells, so we studied that there are 5 types of WBCs. So some of them are granular, some of them are non-granular, right? So neutrophils, eosinophils and basophils. So in short, NEB. Right? So remember this NEB. Neutrophils, eosinophils and basophils. So these NEB are granular. So they are granular WBCs. So they have granules and their nucleus is lobed. Clear? The second one is non-granular or agranular. So they do not have any granules. They are without granules. So cytoplasm does not have any granules and there is single large nucleus. So you will not see any lobes in these nucleus. So I'll show you the diagrams of each and every WBC. So that time you can easily understand. So what are these agranular or non-granular WBCs? So they are LM, that is lymphocytes and monocytes. All these fills, bills, right? So fills, right? So eosinophils, basophils, neutrophils. So all these fills are filled with granules. So their cytoplasm is filled with granules and all the sites, their cytoplasm has no granules, right? So fills have lobes, but sites do not have lobes. Okay, so I hope this is clear. So let us talk about the first WBC, and that is what? That is neutrophils. So tell me, is it a granulocyte or a non-granulocyte? So very clear, fills. Fills are filled with granules. So their cytoplasm has granules and their nucleus is lobed, right? So neutrophils, they are granular. Right, so granular WBCs. Okay, so the nucleus has 3 to 4 lobes. Okay, now the their cytoplasm is granular and they are stained with neutral dyes. So these cells, they are stained with neutral dyes. So around 62% of WBCs are neutrophils. Clear? So as we have studied, their nucleus has lobes, so there are 3 to 4 lobes that you will see. So see, this is basically a nucleus. So one lobe is this, one lobe is this, and one lobe is this. So this is a neutrophil with 3 lobes. Clear? So its nucleus has 3 lobes. So you can see the granules. So these are the granules that are present inside the cytoplasm. So both the things are clear. Okay. So the maximum number of WBCs, what are they? Out of all the WBCs, which WBC is maximum in number? So that is neutrophils. Okay, so they can be stained with neutral dyes, and what is the function? The function is engulfing bacteria. So they perform that role of engulfing the disease causing bacteria. So they perform phagocytosis. And what is the site of production of these neutrophils? So that is bone marrow. Okay, so I hope this is clear. The next one is eosinophils. So eosinophils constitute only 2 to 3%. So also again the phils, which means they are granular. So you can see granules. So these are the granules. And can you see the lobes in the nucleus? So of course we can see these are the 2 lobes in the nucleus, right? So the count increases in allergies. So whenever a person has affected or with any of the allergens, right? So when allergens have been encountered by the body, so at that time the percentage of eosinophils increases. Okay, so this is what a doctor or a pathologist sees in the blood report. Okay, so now what are the features? So the nucleus has 2 lobes, as we can see. There are large cytoplasmic granules, as we can see in the picture. Now they are stained dark red with eosin acidic dye. So neutrophils, right? So as the name suggests, they were stained with the neutral dye. But in case of eosinophils, so they are stained with eosin, which is an acidic dye. Okay, now the functions. The first function is engulfment of bacteria. That is what? That is phagocytosis. So phagocytosis is same in eosinophils and in case of neutrophils. Second is it secretes antitoxins. So we have studied the protection function of blood. So they secrete antibodies or antitoxins. So who does that? Eosinophils does that, right? So they are associated with allergies, as we have seen in the first point. What is the site of production of eosinophils? So again, as in case of neutrophils, it is bone marrow. So eosinophils, they are also produced in bone marrow. Okay, so I hope this is clear. Now the third is basophils. So again, granular with lobed nucleus, right? So it is 0 to 4%. Now what are the features? As you can see, the nucleus is very large and the lobes are not very clear, right? So the nucleus is large, larger are the lobes, and they are not very clear. Then the granules stained with basic dye. So we started with the neutrophils, then we came to what was the eosinophils, then basophils. So the granules were getting stained with the neutral dye, right? Then acidic dye, and then finally when we came to basophils, so B for basophils and B for basic dye, right? And what is this basic dye? So this is methylene blue, and for eosin the acidic dye was eosin only. Clear? So this is the difference. So these are the points of difference between these, right? So remember them. Now the functions. What are the functions of basophils? So they release some chemicals called histamines for inflammation, which dilate blood vessels. So when a person gets affected by certain chemicals, certain allergen, or there is some allergy, so for against those allergens, they release certain chemicals which are called as histamines, which actually dilates the blood vessels. Now the site of production, their site of production is also bone marrow. Okay, now talking about the next WBC, that is lymphocytes. So lymphocytes, so we are talking about agranular WBCs. Okay, so these WBCs, they do not have any granules, so they are around 30%. So the maximum number was of Whom? So 62%. Who was 62%? Neutrophils. Then coming on to little lesser than them, so they are lymphocytes. So they what are the features? So they are the smallest of all the WBCs. So again the question arises, which is smallest of all the WBCs? So that is lymphocytes, right? They have single large nucleus. Now the nucleus is not lobed anymore. So they are lobed only in case of phils, neutrophils, basophils, eosinophils. In lymphocytes and monocytes, the nucleus is not lobed, but there is single nucleus that is large. Now they produce antibodies. Their function is also to produce antibodies, right? So antibodies were also produced by eosinophils. Next is site of production. So they are produced in bone marrow as well as lymph glands like spleen and tonsils. So these are the important points that you have to remember. So make a chart in your notebook and remember them. Okay. Now the last one is monocytes. So again they are agranular, that is non-granular, and they are 5.3% in our blood. So what are the features of these? So they are the largest WBCs. So which are the largest? Largest are monocytes and the smallest are lymphocytes. So basically the largest and the smallest crown has been given to non-granular WBCs. Okay, so remember them, monocytes. The single, right? Mono stands for single. So they are the only ones which are the largest of all the WBCs, and the smallest ones are lymphocytes. Now they have, their nucleus is large and it is, you can see, the kidney shaped nucleus. Okay, at the site of infection they transform into macrophages. So whenever there is a site of infection, they encounter around a site where there is infection, these monocytes, they get converted into macrophages. Okay, so what is their function? To ingest germs. So they phagocytose those disease causing germs, right? Now what is the site of production? The site of production is bone marrow. So almost all of them are produced in bone marrow. Only in case of lymphocytes, they are produced in bone marrow as well as in lymph nodes, right? So in lymphoid organs, right? Lymph organs, which are spleen. Okay, so I hope this much is clear to everyone. So very easily I tried to explain you all of them, and I hope they are clear to you. So just make a chart, as you can see in your book. So just make that chart in your notebook as well and write it in your own hand so that you can understand them better. When you write it, you try to put your attention in that particular chart so you understand it better. So now the origin and life of WBCs. So they are produced in red bone marrow, lymph nodes, and sometimes even in liver and spleen. So most of them are produced in bone marrows. So the average life is about 2 weeks. So average life of these are 2 weeks. So they live only for few hours. As we studied about WBCs, so they live for 120 days. So the RBCs live for 120 days, but WBCs live only for few hours, and about 125 billion neutrophils are produced each day. So old and worn out WBCs are destroyed in the same manner as RBCs. So they are destroyed in the same manner as the RBCs are destroyed. Okay, so the removal of old and worn out RBCs and WBCs is the same. So I hope this much is clear. So now talking about the terms related to WBCs. So there are two terms that you need to know. The one is leukemia. So that is the cancer of tissues forming WBCs whose number increases manifold at the cost of RBCs. So it is fatal, right? So when the tissues which form WBCs, their number increases, so that, and it actually becomes fatal, right? So that increase in count actually becomes fatal for an individual. So that condition is called as leukemia. The treatment is blood transfusion only. So how this leukemia, this cancer can be treated? So it can be treated only by blood transfusion. The next is leukopenia. where there is abnormal decrease in number of WBCs. So when the number of WBCs decreases abnormally, the number decreases abnormally, so that is called leukopenia, and the cancer of tissue producing WBCs, so those tissues which produce these cells, WBCs, so they are that is called as leukemia, right? So these are two very similar terms, very similar kind of meanings, but yet different, right? So I hope you understood the difference between these two. So let us now talk about the functions of WBCs. So WBCs, what do? They protect our body. So that is the body defense mechanism, right? So the very first function is phagocytosis, where they engulf some of the particles. Okay, so when we were talking about the different kinds of cells, different kinds of different types of WBCs, so we spoke about phagocytosis, where WBCs, especially particularly the neutrophils, which are highest in number. How much? So they are around 62% of all the WBCs. So what do? They engulf those particle-like solid substances, especially the bacteria. Correct? So this is a defense mechanism against the disease-causing germs, right? So then, if somebody undergoes a blood test, right, and if the WBC count is more than 50,000 per cubic millimeter, this means that person's body has encountered any infection. So if the abnormal increase in WBC count up to about 50,000 or more per cubic millimeter indicates some infection in the body. So see, this is how those blood reports and the number of cells tell us if there is any infection or the body is healthy, and whatever is the reason. So the pathologist comes to know about the infection by taking a look at the number of cells and the count of these different types of WBCs or RBCs. Clear? So I hope this much is clear. So the first function is phagocytosis. So they engulf any disease causing pathogen. Clear? So I hope this is clear. Let's see the second function of WBCs. The second function is inflammation, which occurs due to the reaction of tissues to any injury or the localized invasion of germs. So these are the two reasons why inflammation occurs, and that inflamed spot has certain characteristics. So they are very common. So if there is any injury to any of your tissues, or there is invasion of any germ into your body, so the first thing that we see is increased local heat. So if this is the place where you got injured, so the heat, you will actually feel heat in that particular area. So you'll see that place will swell, that place will be red, and you'll feel pain also in that particular place. So these are the characteristics of inflamed spot. There are certain leukocytes, especially the monocytes and the neutrophils. So they migrate through the walls of the blood vessels and they reach that particular inflamed portion where they fight against the disease causing microorganisms. And then what do? They phagocytose. The first function of WBCs, right? The phagocytosis. So they phagocytose the disease causing organism. So this is how they fight against those infections. Also, pus is mainly composed of dead WBC. So sometimes you see that pus, right, a white, you'll see a swelling, and it seemed to be something white fluid inside that area, right? So that is pus, and what it is composed of, that pus is usually composed of dead WBCs together with the tissue cells destroyed by the bacteria. So that pus is all because of those dead tissues, right? So bacteria has invaded, so the tissues of that particular area are dead, and then WBCs have come and they have engulfed those bacteria. So pus is all due to the dead WBCs together with the tissue cells. Clear? So I hope this much is clear. Third function is formation of antibodies, or you can say antitoxins, right? So the lymphocytes, they produce antibodies. So these lymphocytes actually produce antibodies which kill or neutralize the germs or poisons from them, right? So the lymphocytes of our body, they play this role of producing antibodies. So whenever our body encounters any of the germs, so our lymphocytes, the cells in our body, they produce antibodies to fight against those infections. So the vaccination, so you must have heard about vaccination. So it is a very common word after COVID, right? So introducing weakened germs or germ substances, right? So their protein, either you take a dead microorganism, that pathogen basically, or weakened pathogen, right? Or you take any part of that microorganism. So these are introduced into the body of the person, right? So as soon as our body encounters any foreign particles, so our immune system becomes active, and they stimulate the formation of particular antibodies which will destroy these particular disease causing germs. So this is how WBCs, they fight against antibodies. Clear? So I hope the function of WBCs are clear to you. So as we were talking about antibodies or antitoxins, so the disease causing germs, when they enter into our body, they release some poisonous substances which are called toxins into our bloodstream. So the lymphocytes, these are the WBCs that are present inside our body. So they produce antibodies against these toxins, right? So we can also call them as antitoxins. Clear? Now the antibodies, they are toxic specific. So whatever the antibody is produced by our body, they are specific to that particular toxin and act as antitoxin which neutralize or detoxify the poisonous effect of that toxin, right? So soon after a person gets infection, so the level of these antibodies in our blood, they increases manifold, and also they do not go down, the count does not go down in as such, but you will observe if that person undergoes a blood test, so that person's blood will show antibodies for a very longer period of time, right? And this is the underlying principle of vaccination. So vaccination is based on this only. So as soon as the toxin enters into the body, our body produce antitoxins, right? And these antitoxins act on the toxin, they neutralize, they nullify or detoxify the effect of these toxins, and finally our body gets recovered from that particular toxin. So these antitoxins or these antibodies, they are specific for these toxins. So our body produces specific antitoxins for a particular toxin, and once our body get encountered by any of the toxins, so then our immune system keeps a memory of it. So our immune system just keeps the memory, and in future whenever there is further subsequent exposure to the same pathogen, so our immune system already know that yes, the body has encountered. So this is all because of the memory of the immune system, right? And the immune system starts making those antibodies which will destroy that particular toxin. Okay. Now the next is blood platelets. So these are the third type of cells which are found in our body, and we also call them as thrombocytes, right? So these are very minute bodies. These are oval or round in structure, and they are non-nucleated. So RBCs were also non-nucleated. They do not have nucleus, but all the WBCs have nucleus, right? Now talking about platelets, so they do not have nucleus as well, and they float in the blood. So if we talk about the number of platelets in our body, so they range from 2 lakh to 4 lakh per cubic millimeter of blood in an adult human. So they are derived from giant cells in the bone marrow, and these cells are named as megakaryocytes. So how are these cells derived from them. So basically these are the cells from where these small little platelets, right? So they bud off from the megakaryocytes in such a manner that each of the platelet is surrounded by a membrane, and their life cycle is very less. So their lifespan is very less, that is just 3 to 5 days. The abdomen, and finally when they die, so they are destroyed in spleen, which is one of the lymphatic organ which is situated in our abdomen, right? So how are they similar to our RBCs? So RBCs are enucleated, right? So they are, when the RBCs are produced, they have nucleus, but soon they mature, they lose that nucleus. So same is the case over here. Platelets do not have any nucleus, right? Then they are produced in bone marrow as well. And from which cells they are produced? So they are produced from megakaryocytes, correct? And they live for 3 to 5 days only. Okay, and they are also destroyed in spleen. Now blood platelets, they are called as initiator of blood clotting. So we already know that which cells of the blood play very important role in blood clotting. So they are blood platelets, or we call them as thrombocytes. So they are the initiators of clotting of blood. So when there is a site of injury, when a blood vessel gets injured, so platelets, they disintegrate and they release a chemical substance which is called as thrombokinase, which initiate the process of blood clotting. So just take an overview of how this the blood clotting happens. Okay, so if you see, this is a blood vessel, right? So inside the blood vessel there are different types of cells. So there are RBCs, there are platelets, and there are another cells also. So as soon as the blood vessel gets damaged, so all the platelets will come to the damaged site and they will form a platelet plug. And finally the fibrin also comes here and they form the thread. So thread of the fibrin trap RBCs and they form a clot, right? So this was just an overview how clotting happens in our body. So let us go into the detail of this clotting. Now the clotting of blood is also called as coagulation of blood. So let us see how does that happen. What are the main reactions in blood clotting? So whenever you see an injured tissue or injured cell, right? So at that place, the platelets will come and they will release a factor called as thrombokinase. So it has some other names also, thromboplastin, factor X, or Stewart factor, right? So these are the different names which are given to thrombokinase. So thrombokinase is basically an enzyme which helps in clotting, which is released by platelets. And now this thrombokinase will actually go and it will activate prothrombin. So prothrombin is actually an inactive thing which is found in the blood plasma. Okay, so it is inactive in blood plasma. So the thrombokinase, along with the calcium ions which are again present in the plasma, they will activate this prothrombin into thrombin, which is an active form. Okay, for the production of prothrombin, vitamin K is very, very important. So if you want your body to prepare prothrombin, which is very important in clotting of blood, you should eat more and more of vitamin K, correct? So then prothrombin will get converted into thrombin. Now in the next step, this fibrinogen, which is a soluble form in blood plasma. So this fibrinogen, in the presence of thrombin along with the calcium ions, they convert this fibrinogen into fibrin, which is insoluble, right? Now these fibrin, it is very sticky, right? And it forms a mesh-like structure, right? So it forms a network. So these are very microscopic threads which form a mesh, right? So they form mesh at the site of blood clot, and they form a network of sticky threads, and plus the RBCs, they will form a clot that is also called as thrombus. So I hope how the blood clotting actually happens is clear to you. So these fibrins, these prothrombin, fibrinogen, all of them are very important if we talk about blood clotting. So the calcium is very important. So if you want your body to form clots very quickly as soon as you get even a small cut, so what you will have to do, you will have to take in lot of calcium, you will have to take in lot of vitamin K, right? So I hope the main reactions of blood clotting are clear to everyone. So if we talk about the fate of blood in clotting, so what happens? There is blood, and now this blood is composed of two things. One is plasma and one is corpuscle, so which means cells, right? So this plasma is composed of fibrin or serum, right? So there are two things from which this plasma is composed of. One is fibrin, which forms that mesh-like structure, the microscopic threads which form a sticky mesh-like structure which prevents the further blood loss, right? So plasma is composed of serum and fibrin. So cells along with the fibrin, so these cells of the blood along with the fibrin, they make a clot which is called as blood clot, or it is also called as thrombus, right? And now as soon as this mesh-like structure, it squeezes, it becomes, it gets squeezed, so it releases out some liquid, right? So that liquid that is coming out of that clot is actually serum. And what the solid part that is left behind, which contains fibrin and the cells, is called as clot. Okay, so I hope all the complete process of blood clotting is clear to everyone. Blood clotting is even possible inside a small test tube. So if we take out the blood and if we put it in a test tube, so after some time, what you will observe that the serum and clot has separated. So this is the serum, the yellow portion is the serum, right? These are the fibrin threads. If you get into the depth of the blood, and these are the different types of cells which are present, right? So you keep it for some time. So after some time you will absorb the blood and basically the cells, and this fibrin threads, they will form a clot. What do? They will form a clot, right? And the rest, the serum will get separated. So the clot will settle down and the yellowish color fluid, the serum, will get separated from the clot. So this is even possible if there are some rough surfaces inside the blood vessels. So you must have seen whenever the cholesterol level of any person, you must have heard basically. So whenever the cholesterol level of any person increases, so that roughness inside the blood vessels, right? So that leads to the formation of clot in the blood vessels. So the blood clotting this experiment can be performed very easily in laboratory also. There is one disease which is related to blood, and it is a genetic disorder. So that disease is Haemophilia, which is a genetic disorder in which blood does not clot. So that is a genetic disease, right? So you must have heard about the chromosomes, that there are autosomes and there are sex chromosomes. So this disease is linked to the sex chromosomes. So the gene is present on the sex chromosomes. So the blood does not clot because there is the loss, there is a lack of blood clotting protein. So the blood clotting proteins are absent in the body, which leads to this disease. So it is a genetic disease. So it is transferred from parents to the offsprings. So in case the platelet number goes down, the coagulation occurs very slowly and it often leads to hemorrhage. So if we know that platelets are very, very important if our body has to form the clot, right? So for the formation of clot, if you say the number of platelets in the body, they go down. So what will happen, even if there is a small cut, so that small cut will not form a clot and there is a loss of lot of blood, which will lead to hemorrhage in the body, and it occurs in many diseases. For example, viral dengue fever. So in 1996 in India, in Delhi and in many other states, so this fever was, this was a viral fever that was spread in 1996, which led to the death of many people because their body was not able to form the clot. right? So it is very important to keep yourself safe from the mosquitoes during that rainy monsoon season, because those mosquitoes, they spread dengue, and in this viral fever, the number of platelets, they come down drastically, and when this comes down drastically, there are the chances that the blood clotting will not happen, and there might be hemorrhage in that person. There might be lot of bleeding inside that person's body. So this is one of the genetic disorder which is related to blood. So let us talk about some of the questions now. So the first question is squeezing out of WBCs, white blood cells, from the capillaries into the surrounding tissue. So what is the term used for this particular statement? So that is diapedicels, right? So you remember the diagram where we have studied that the shape of a WBC is amoeboid. It forms pseudopodia and it squeezes out of the blood vessels into the capillaries, into the narrow capillaries. So we give it a term diapedicels. Clear? Now the next question is non-granular WBCs are. So there are WBCs, right? And we have studied there are of two types. One are granular and the other ones are non-granular. So all those who are filled, they are filled with granules, right? So their cytoplasm is filled with granules. So neutrophils, eosinophils and basophils, NEB, they are granulocytes, whereas lymphocytes and monocytes, they are non-granular. Their cytoplasm does not have granules. So what is going to be the correct option over here? So A is going to be the correct option. Next question is WBCs engulf bacteria in the process called. So the WBCs, they engulf bacteria. So what do we call that process? So that process is it diapedicels? No, it is squeezing out of the WBCs, right, from the blood vessels. So this is wrong. Is it phagocytosis? So that is correct. So when these WBCs, they engulf any bacteria or disease causing germ, so we give it a term as phagocytosis, right? So active and passive transport, we studied them in plants, right? Physiology of plants. The next question is give reason why mature mammalian erythrocytes, so we are talking about RBCs, lack nucleus and mitochondria. So we studied it in very detail. So there is one more organelle that is absent, that is endoplasmic reticulum. So since these mature erythrocytes, they lack mitochondria and nucleus, so that they can accommodate more hemoglobin, and this hemoglobin can accommodate more oxygen molecules. So more oxygen molecules can be carried by the RBCs. So lack of such organelles provide a peculiar biconcave appearance to RBCs, which aids in efficient diffusion or efficient transport of gases, right? So I hope this question is also clear. Now the next question is study the following diagram carefully and then answer the questions that follows. So these are the diagrams. So the first diagram shows a blood vessel where there are lots of cells, right? And one cell is getting squeezed out of that blood vessel, right? So let us see what are the questions. Name the cell labeled 1. So what is this cell labeled 1? So it is almost red in color. So which cells are red in color? So they are red blood cells, right? Or you can say erythrocytes. Identify the phenomena occurring in A. So what is this phenomena which is occurring in A? So this phenomena is diapedicels, right? That is squeezing out of WBCs from the blood vessel. Now the next is mention the two structural differences between 1 and 2. So 1 and 2. So this is 1 and this is 2. So it means we have to tell the structural differences between WBCs and RBCs. So the RBCs, they are enucleated. So they do not have nucleus, whereas WBCs, they have nucleus. Correct. So this is one of the difference. What is the second difference between WBCs and RBCs? So we have to talk about the structural difference. So what is the difference in their structure? Okay. So RBCs, they have disc shape, right? Disc shaped. They are biconcave. So this was the first difference, this was the second difference, right? So over here, the WBCs, they are usually round, or they are oval, or they are of different shapes, right? Okay, but they are not biconcave. The D part is name the process occurring in B and C and state the importance of this process in human body. So what is happening in B and C? So in B and C, this particular WBC is phagocytosis. So what is happening over here? So phagocytosis is happening, where this WBC is engulfing all those disease causing germs, disease causing bacteria. And why is it important? It is important as a defense mechanism of our body. So our body is getting protected against these disease causing organisms. Okay. So I hope the blood vessels, the RBCs, WBCs and platelets are clear to everyone. So now we will meet in our next video, where we will discuss the rest of the chapter. So till then, take care, and as you know, learn to have free hair पर best है.